Okay, so please go ahead with the discussion. You have 11 minutes.
Assalamu alaikum. I am Abdullah Numan and my supervisor is Dr. Muhammad Saifur Rahman. The title of my thesis is A Novel Semi-Supervised Method for Segmenting Spatial Transcriptomics Data. So let us start with the spatial transcriptomics technology. There are a lot of spatial transcriptomics technology, but among them, the famous one is the VCM technology by 10x Genomics. In this technology, in each VCM slide, there are 6 capture areas. In each capture areas, there are 5,000 barcoded spots. From those spots, we get gene expression from the gene from all the genes expressed in that spot. After the sequencing, we get 3 things. The first thing is a gene expression matrix that represent the count of each expressed gene. We also get the spatial coordinate of the genes that represent the spatial position of the tissue of the genes of the spots in the tissues. We also get a very high resolution histology image. Now we will discuss the motivation for the topic. So first of all, the recent trends in spatial transcriptomic methods have been shifted to spatially resolved transcriptomics. Previously, the famous method was single cell sequencing, but now the shift has been toward the single cell multiomics and spatially resolved transcriptomics that has attracted many researchers across the world. So that has been an important motivation. And the important thing is that spatial transcriptomics can be used to spatial domain detection in the disease tissues. If we can detect different regions in our tissue, we can make targeted therapy for that for different cells that will be very useful for us. Here we can see a melanoma cancer tissue where the black contour represent the melanoma affected cells, and the yellow regions represent the lymphoid tissues, and the red regions represent the stroma. Melanoma is a cancer tissue, so the black contour represents that cancer, and the lymphoid tissues represent the white blood cells that are grown around the cancer tissue, and the stroma tissue represents the cells that promotes the growth of the cancer tissue. So if we can identify those regions correctly using our analysis, that will be very helpful for targeted therapy for different patients. Now we will discuss the related works in this domain. Most of the work done in this domain are unsupervised. So first of all, we will discuss the M-CLUST method. This is a very popular method. This uses a Gaussian mixture model. For estimating the parameters in this model, EM algorithm is used. But the problem in this model is that this does not incorporate the spatial information among the spots. So the tissue clustering is not very continuous. Another very famous method in this domain is Bayes space. Bayes space uses Bayesian model with Markov random field. This Markov random field a priori is used to get the spatial information. This is ensured by the Potts model. Bayes space estimates the parameters for clustering using Monte Carlo Markov chain simulation. Another very famous method in this domain is SCMEB. SCMEB also uses the spatial information using a hidden Markov random field. It estimates the parameter using ICM-EM algorithm that finds the parameter. Here we saw that any of those methods did not use the human inputs here. But can we use the human input for improving our clustering? That is a burning question. So we have developed a methodology that incorporates the human experts scribble that can be useful for us. So first of all, we did data processing. In this processing step, we took 2000 highly variable genes from all the genes. In each spots in DLPFC dataset, in each spot there are 35,000 to 40,000 genes. Though so many genes are not very easy to work with, so we only take the highly variable genes. After that, we have used a principal component analysis to reduce the dimensionality of the data so that there is minimum loss in the information and maximum information is captured in a lower dimension. After that, we have to take scribbles from expert annotators who can give some scribbles in the tissue histology image. Here we can see two scribble. One is blue that indicates a tumor region, and orange scribble that indicates non-tumor region. So now let us formulate the problem. Our methodology is inspired from the image segmentation algorithm from Kim et al. This algorithm takes takes an image as input and makes an segmentation on the image. That is, each pixels in the image is segmented into a group that identifies a object. We can model our problem into this by thinking the pixel of the images as different spots and identify different region in the tissues using an existing image segmentation algorithm. So to formulate the problem, we have to think of three components. One is similarity component that identifies the similarity in gene expressions among spot. The continuity component that takes care of the spatial correlation among the neighboring spot, and the scribble component that takes care of the agreement between the scribbled spot and the predicted cluster by the algorithm. To solve the problem, we have taken the model architecture. We have shown the model architecture here. Our model takes as an input the principal component of the gene expression feature. Those gene expression features are passed through the one-on convolution, the inception block, and another one-on convolution to find a output response map that gives the probability of each spot being in a cluster. Doing a argmax, we get the spot level for this epoch, and with that we calculate the similarity loss between the spots. We also compare the spot level with the scribble to find the agreement between the scribble and the spot level. With this, we get a scribble loss. Combining those two loss, we get the final loss component, and this is used to back propagate and train the model. In this model, we have used the inception block that captures the continuity portion of the problem. In inception block, we have used different type of kernels that are 3 into 3, 5 into 5, and 1 into 1 convolution that captures varying resolutions that handles the continuity part of our model. For the similarity loss, we have used a cross entropy loss that models the loss between the output response map and the output spot clustering. We also used a cross entropy loss to formulate the scribble loss that handles the agreement between the scribble and the output spot level. The total loss is computed as alpha into L sim and added by 1 minus alpha into L scribble. Here alpha is the parameter for our alpha is a hyperparameter. So that was our part for expert scribble option. where we have an scribble from an expert annotator who has identified different regions. But if we do not have the scribble on hand, we can automate the scribble using a backbone approach, in which we take output of a non-spatial clustering method and find a backbone find a backbone using an algorithm in which we see each spot, and if all the neighboring spot is of same level, we take that spot in our backbone. But the problem here is that non-spatial algorithm does not provide as good result as expert scribble, for which we have to randomly drop some spots in every scribbled spot in every iteration, so that we can learn, our model can learn on its own without the impact of scribble in those spots. This dropout rate is also controlled by a parameter that is called beta. To fine-tune the parameters, we have done the grid search on those parameters. We have two parameters, that is alpha and beta. In alpha, we moved over 0.5 to 0.95.
You are over. Please wrap up in 1 minute.
In beta, we have moved over from 0.3 to 0.5 with an interval of 0.5. We chose an hyperparameter that maximized the log likelihood score. To show the result, we have to show the melanoma dataset, in which from the expert scribble, we was able to detect the melanoma lymphoid regions and the stroma portion more accurately than the base space. We get an ARI of 0.75 that are we get an ARI of 0.86 that are way higher than the ARI given by base space that is 0.75. In unsupervised setting also we get a good result, though the ARI is a bit low here, but we qualitatively it is better than that of the result of the base space, since we can identify the melanoma, we can identify the lymphoid tissues more accurately.
On human, you are way over. 30 seconds.
I have 4 slides.
30 seconds conclusion.
Inshallah. In the human DLPFC dataset, we also perform good with expert scribble. Our ARI is better in many samples. The result is shown with a violin plot here. The mean ARI of our using our method in expert scribble is very high, On breast cancer dataset with